The word gene originates from ancient Greek meaning locus or region. The concept of the gene originated with the work of Gregor Mendel and his discoveries of inheritance patterns. While he never used the term gene, he developed the basics of its modern context. While looking at the inheritance of a variety of physical characteristics of pea plants, such as flower color, seed shape, etc., he discovered that the inheritance was best explained by the presence of discrete, heritable units. The widely accepted inheritance hypothesis of the day was known as blending inheritance, which predicts that the traits of the parents blended in the offspring. For example, a white flowered plant and a purple flowered plant would produce an intermediate or light purple flowered plant. To test this hypothesis, Mandel carried out a series of experiments on the common pea plant in his monastery's experimental garden plot between 1856 and 1863. The pea plant awarded Mandel several advantages for studying inheritance. First, pea plants grow easily and quickly, allowing for many replicates and generations in a relatively short amount of time. Second, peas have many different physical characteristics or phenotypes. Mandel eventually analyzed 7 phenotypes: seed shape, seed color, flower location, pod shape, pod color, and plant height. Most importantly, he could obtain true breeding varieties or pure lines. When these seeds were planted, they produced known phenotypes. For example, pure line seeds for purple flowers would always develop into plants with purple flowers. Pure lines are produced by self-pollination, where a flower is covered and the pollen of a plant is used to fertilize the same plant, essentially producing a clone. Therefore, whatever phenotypes the self-fertilized plant had, the seeds would produce plants with exactly the same physical characteristics. These true breeding pure lines are known as the parent generation, or pea generation. Mandel initially focused his analysis on seed shape. Pea seeds are either round, smooth, or have angular indentions, wrinkly. After self-pollination, he grew out pure line seeds of both varieties until they flowered. To control against unwanted pollination, the developing flowers were covered with mesh bags. Once fertile, pollen was collected with a sterilized paintbrush from the flowers of one phenotype, such as smooth seeds, and used to pollinate the opposite phenotype, such as wrinkly seeds, producing a hybrid of the pure lines. He also cross-pollinated smooth-seeded varieties with wrinkly-seeded varieties too. Once pollinated, the mesh bags were reinstalled and the seeds allowed to develop. These hybrids were notated as the first filial generation, or the F1 generation. While it was known that crossing similar true breeding plants would produce the same phenotypes, or pure lines, the blending inheritance hypothesis predicted that crossing 2 different pure lines would produce an intermediate in the F1 generation. Mandel expected to get an intermediate result of slightly wrinkly-seeded plants from a cross of pure line smooth-seeded plants with pure line wrinkly-seeded plants. Mendel's results conflicted with this prediction. Once the seeds developed from Mendel's monohybrid cross, he discovered that all the resultant seeds, the hybrids, were not semi-wrinkly. Rather, they were identical in shape to the round-seeded pure lines. From his controlled pollination, he knew that the hybrids had equal components of the wrinkly-seeded and smooth-seeded parent pure lines. He concluded that the smooth seed phenotype masked the expression of the wrinkly-seeded phenotype. He termed the expressed phenotype of the hybrids, smooth seeds in this case, as the dominant phenotype, notated as a capital letter, for example, capital A. Whereas the repressed phenotype, wrinkly seeds, was termed the recessive phenotype, notated as a lowercase letter, for example, lowercase a. This result directly contradicted the blending inheritance hypothesis. Hybrids were not intermediates. From this initial study, Mandel concluded that the physical characteristics or phenotypes he was studying, such as seed shape, flower color, etc., were determined by the combination of what he referred to as particles, which he defined as discrete heritable units. This concept, known as the particulate inheritance hypothesis, was in direct contrast to blending inheritance, which predicted that the phenotypes of offsprings are intermediate to the parents. We now refer to Mendel's particles as alleles, alternate versions of a gene. Mandel discovered the presence of dominant and recessive alleles. Furthermore, he predicted that an offspring receives exactly one allele from each parent, giving each individual two alleles. The combination of those alleles is known as an individual's genotype, of which there are three possibilities. Two dominant alleles, termed homozygous dominant, notated with two capital letters, for example, capital A, or two recessive alleles, termed homozygous recessive, little a, or one of each allele, termed heterozygous, capital A, little a. From his monohybrid cross, Mandel predicted that both the homozygous dominant, big A, and the heterozygote, big A, little a genotypes would produce a dominant phenotype, for example, round seeds. While the recessive phenotype, wrinkled seeds in our case, is only expressed by the homozygous recessive genotype, little a. This became known as Mendel's principle of dominance, where a genotype is composed of two alleles, and if the alleles differ, only one phenotype will be expressed, the dominant one, while the other is masked, the recessive one. Blended inheritance predicts that the heterozygote would be an intermediate between the two homozygous phenotypes. After this discovery, Mendel grew out the F1 generations, the hybrids of the pure lines, and then repeated the same cross-pollination, creating new seeds. These seeds form the second filial, or F2 generation, which can be thought of as the hybrids of the original hybrids. To his surprise, not only did the dominant genotype round seeds emerge in the F2 generation, but also the recessive phenotype, wrinkly seeds. More precisely, he collected 5,474 round seeds and 1,850 wrinkly seeds, or a 2.96 dominant to 1 recessive ratio, approximately 3 to 1. Mendel then repeated the same experiment for a variety of other traits with his peas, such as seed color, flower color, and pod shape. From his original experiment, he predicted that in each of the physical characteristics, one phenotype would dominate in the F1 generation, masking the recessive phenotype. From his seed shape experiment, he also predicted an approximate 3 to 1 ratio of dominant to recessive phenotypes in the F2 generation. And that is exactly what he found. Let's use seed color as an example. Mendel crossed pure line yellow-seeded and green-seeded plants. Seeds from that cross, the F1 generation, were all yellow, indicating the dominant phenotype. After growing those seeds out and crossing the hybrids, he counted the F2 seeds: 6,022 yellow and 2,001 green, a 3.01 to 1 ratio. He found a similar ratio for the other characteristics in the F2 generation. Pod shape was 2.95 inflated to 1 constricted, pod color was 2.82 green to 1 yellow, and flower color was 3.15 purple to 1 white. All approximately 3 to 1. Mendel's discoveries went largely discounted and forgotten due to a strong acceptance of a competing inheritance hypothesis. More on that later. At the turn of the 20th century, researchers designed experiments that led them to the same conclusions Mendel arose at decades earlier, without knowing of Mendel's work, we think. Eventually, his publications were rediscovered by geneticists, and he was given due credit. One of those scientists was Reginald Punnett, who developed a simple tool now known as the Punnett square to predict the probability of genotypes and phenotypes from controlled crosses. Let's revisit Mendel's monohybrid cross of flower color using a Punnett square to visualize Mendel's predictions. From the results of his F1 generation, Mendel determined that the purple phenotype for flower color was dominant, while white flower was recessive. Based on his principle of dominance, purple flowers could either be homozygous dominant F, or heterozygous F. And white flowers were homozygous recessive f. When Mendel self-pollinated the pea plants to produce a pure line for the pea generation, he was creating a homozygote, either big F, or little f, depending on the original plant. Once the pure lines reached reproductive maturity, he cross-pollinated a homozygous dominant with a homozygous recessive, notated as big F times little f, little f. Once the seeds matured and were allowed to develop into plants, the F1 generation, they all expressed the purple flower phenotype, but were known to be hybrids, capital F, little f. He concluded that one of the dominant alleles from the homozygous dominant parent and one of the recessive alleles from the homozygous recessive parent were both present in the plants of the F1 generation. In other words, all plants in the F1 generation were heterozygotes and expressed the dominant phenotype for flower color, purple. Next, he crossed the plants of the F1 generation, big F little f times big F little f, producing offspring that developed into the F2 generation. To calculate the predictions of this cross, we use a Punnett square. Mandal predicted that there was an equal probability of an offspring receiving either allele present in the parent. If a parent was heterozygous, big F little f in this case, each offspring would have a 50% chance of getting a dominant allele, big F, and a 50% chance of receiving a recessive allele, little f. In the Punnett square, this is visualized by separating the alleles of one parent and placing them above a box bisected vertically. If the other parent is also heterozygous, the box is also bisected horizontally, and the alleles of the other parent are placed one on top of the other to the left of the box, representing the equal probability of receiving that parent's alleles. The four boxes in the middle represent the probability the offspring will acquire a specific genotype. Once the parental alleles are in place, transcribe the top alleles down into each internal box below it and the side alleles to both boxes to its right. In this case, we end up with one box that is homozygous dominant, big F, two boxes that are heterozygous, big F, little f, and one box that is homozygous recessive, little f, little f. With this tool, we can predict the probable genotypic ratio of the offspring, 25% big F to 50% big F, little f, to 25% little f. Simplified as 1 big F to 2 big F, little f's to 1 little f. However, Mandal could not see the alleles, but could detect the phenotypes. Since both big F, and big F, little f appear purple, and little f appears white, he could predict the phenotypic ratio, 75% purple-flowered plants to 25% white-flowered plants, or 3 purple to 1 white. This explains the approximate 3-to-1 ratio he detected among several different phenotypes. The fact that Mandal observed this consistent 3-to-1 phenotypic ratio in the F2 generation across several different traits allowed him to expand the particulate inheritance model. In what has become known as Mandal's principle of segregation, Mandal predicted that an organism's physical traits are passed from parental generations as discrete particles, now referred to as alleles, with exactly one particle from each parent in each gamete, for example, pollen or egg cells. Therefore, each gamete has an equal probability of receiving either allele present in the parent. When an organism generates a gamete, only one of two possible parental alleles is allocated to each gamete. Furthermore, the allele allocation is random, which explains why there's an equal probability of receiving either allele. When gametes fertilize, a new organism is formed, which have genotypes that are a combination of parental alleles. After Mandal discovered the principles of segregation and dominance using his monohybrid crosses, the next big question was whether or not the inheritance of multiple phenotypes are linked. There are two alternative hypotheses, dependent and independent assortment. The dependent assortment hypothesis predicts that the inheritance of physical characteristics are linked during gamete formation. For example, an allele that codes for a specific flower color is connected to an allele for seed color. Alternatively, independent assortment predicts alleles for different phenotypes are sorted into gametes independently from one another. In other words, receiving an allele for one phenotype, such as flower color, does not determine the presence of another phenotype, such as seed color. To test these competing hypotheses, Mandal conducted a dihybrid cross. He identified pea plants that had dominant phenotypes for two characteristics and self-pollinated them. The plants resultant from those seeds were pure lines for two dominant characteristics. In this case, the pure line pea plants for the pea generation would be homozygous dominant for purple flowers and homozygous dominant for yellow seeds. These plants are notated as big F, big S, big S. The same procedure was conducted with plants that had the recessive phenotypes for both characteristics, producing pure lines with white flowers and green seeds, little f, little s, little s. Next, the P generation was crossed and grown out, producing the F1 generations of dihybrids, all big F, big S, little s. As expected, all of the dihybrids express the dominant phenotypes, purple flowers and yellow seeds. When the F1 generation reached maturity, he conducted a controlled cross of the dihybrids and grew them out, producing the F2 generation. The competing alternative hypothesis predicted different results for the F2 generations. If alleles were linked as predicted by dependent assortment, the big F allele would be physically connected to the big S allele in the production of a gamete, and the little f allele and the little s allele would be linked in another gamete. Therefore, in a dihybrid cross, the pollen and eggs would either have an allele combination big F and big S, or little f and little s. No other combinations are possible, not big F little s or little f big S. If dependent assortment was supported by this experimental design, the predicted genotypic ratio would be 1 big F, big S to 2 big F, little s to 1 little f, little s. And the phenotypic ratio, what he could actually observe, would be 3 purple flower, yellow-seeded plants to 1 white flower, green-seeded plants, which can be generalized as 3 dominant to 1 recessive for other phenotypic combinations under investigation. Dependent assortment would not produce plants with a combination of dominant and recessive phenotypes from a dihybrid cross. In other words, there would be no purple-flowered green-seeded plants, nor would there be white-flowered yellow-seeded plants. In contrast, the independent assortment hypothesis predicted that all allele combinations were possible in gamete production. Big F, big S, big F, little f, big S, and little f, little s. This expands the Punnett square from 4 boxes to 16. The prediction of the phenotypic ratio of independent assortment in this case would be 9 purple-yellow to 3 white-yellow to 3 purple-green to 1 white-green. Generalized as 9 dominant to 3 dominant recessive to 3 recessive dominant to 1 recessive recessive. Results of his dihybrid cross were consistent with the independent assortment, indicating that alleles from different phenotypes segregate independently of each other. This hypothesis became known as Mandal's principle of independent assortment. Mandal's elegant experiment clearly disproved the blending inheritance hypothesis. He proposed an alternate hypothesis, the particulate inheritance hypothesis, which has 3 main components. First, the principle of dominance predicts that each phenotype is determined by 2 alleles, one of which is expressed when present, known as the dominant allele. Recessive phenotypes are expressed only when an organism is homozygous recessive. Second, the principle of segregation predicts that during gamete production, each gamete receives exactly one of the 2 alleles present in the parental cell at random. And third, the principle of independent assortment predicts alleles coding for different phenotypes are segregated independently from one another during gamete formation. Mandal's success was in part due to the pea plant's ability to self-pollinate, creating pure lines. However, nearly all animals are incapable of self-fertilization. This makes understanding the genetics of animals like humans increasingly more complicated than plants. Pedigree analysis is a method that uses the principles developed by Mandal to look at families' genetic histories with an attempt to understand how phenotypes are expressed. In a pedigree, squares represent males and circles represent females. Horizontal lines represent a mating event producing children. Children are connected to their parents' horizontal line by a vertical line and are shown below their parents. Generations are shown with Roman numerals such as 1, 2, 3, etc. Pedigree analyses are typically used to follow a specific genetically inherited trait, which is customarily shaded. If 2 parents have the same phenotype and produce a child that has a different phenotype, we know that the child's phenotype must be the recessive expression. Furthermore, we know that the child's genotype must be homozygous recessive, for example, little a, and the genotypes of both parents must be heterozygous. Using this logic, we can begin to deconstruct larger pedigrees and predict its members' genotypes based on the relationships with individuals containing the recessive phenotype. In this example, all the members with the genetically inherited trait are homozygous recessive. Each individual that is homozygous recessive received exactly one recessive allele from each parent. Therefore, if a child has a recessive phenotype and both parents have a dominant phenotype, those parents must have been heterozygous. If we know that the parents are heterozygous, we cannot determine the exact genotype of a child that expresses the dominant phenotype without further information. He could either be homozygous dominant or heterozygous. His genotype is denoted as big A question mark. The same is true for the original parents of the third child. If the parents have both the dominant and recessive phenotype, and their child has a dominant phenotype, we know that the child's genotype is heterozygous because one recessive allele must have come from the parent with the recessive phenotype and the dominant allele from the parent with the dominant phenotype. The same logic holds for this couple's other child and the remaining individual. Pedigree analysis is a useful tool in genetic counseling and understanding human inheritance. Mandel's discoveries went largely unnoticed or ignored due to the strong acceptance of the competing inheritance hypothesis within the scientific community, blending inheritance. Acceptance of the particulate inheritance model was also very likely discounted due to his ignoble status among the academic elite of the day. At the time of Mandel's publication in 1866, the major topic of conversation in the biological community was Charles Darwin's theory of natural selection, as described in On the Origin of Species, published in 1859. While the theory of natural selection was gaining greater acceptance, Darwin published his own theory of inheritance known as pangenesis in 1868. In his description, he suggested that all organs of the body contain unseen gemmules that are extracted and travel to the reproductive cells. In this manner, a reproductive cell would consist of many gemmules from all organs and tissues of the body. In humans, for example, sperm and egg cells would acquire particles from all the parts of the body, the heart, skin, feet, etc. And during fertilization, these gemmules would mix, forming a new organism. Darwin's pangenesis theory was an extension of his grandfather's beliefs. In 1794, Erasmus Darwin in Zoonomia suggested that animals have arisen from one living filament, with power of acquiring new parts, based on the animal's interactions with its environment, and each acquired acquisition is inherited by successive generations. In his widely acclaimed book, Philosophie zoologique, in 1809, Jean-Baptiste de Lamarck expanded this concept, creating the first formal theory of inheritance, the inheritance of acquired characteristics. Essentially, Lamarck suggested that individuals acquire physical characteristics during their lifetime through their interactions with the environment, and those acquired characteristics were inherited by their offspring. The giraffe's neck is a classic example. Lamarck argued that giraffes stretch their neck to reach higher food sources, eventually leading to a longer-necked individual. He posited that this acquired characteristic was passed to the next generation, a trend which continued in successive generations, eventually leading to the long-necked giraffes we see today. Mandel's study was diametrically opposed to Darwin's pangenesis theory, which supported both the inheritance of acquired characteristics and blended inheritance, even though Darwin had no experimental analyses to support these concepts. Furthermore, Mandel rejected Darwin's concept of natural selection, arguing that species are fixed and do not change over time. While Mandel's work was known about by his academic contemporaries, his conclusions were essentially neglected as a result of a lowly monk's clear opposition to the biological superstar of the day, Charles Darwin. After several decades, Mandel's laws were rediscovered by Walter Sutton and Theodore Boveri in 1900, leading to a widespread rejection of Darwin's pangenesis theory. The rediscovery of Mendelian genetics, combined with advances in microscopy and experimental design, allowed Sutton and Boveri to establish a more accurate genetic model known as a chromosome model of inheritance. And this is the topic of our next chapter.